Today, we will be presenting on human milk and lactation during the COVID-19 crisis. Our first presenter is Dr. Lars Bode. He will be presenting on breast milk in the time of contagion. Dr. Bode is a professor of pediatrics at the University of California in San Diego. He's a director of the Larsson-Rosenquist Foundation Mother Milk Infant Research, a Center of Research Excellence, and past president of the International Society for Research in Human Milk and Lactation. So without further ado, I'm going to turn things over to Dr. Bode so we can begin his presentation.
Thank you, Laura, and thanks for inviting me to present some of the data that we collected over the last 6 months, specifically on SARS-CoV-2 and COVID-19. So before I start, a quick slide on disclosures, where most of our funding is coming from. This is independent of the work that I'm going to present today, but most of our funding is coming from the NIH, the Gates Foundation, the Larsson-Rosenquist Foundation, and I do get travel reimbursements and honoraria from different societies, foundations, company to speak at events like these. Okay, I'm preaching to the choir here, but I think it's very important to understand that there is quite a bit of work that has been done before this pandemic hit. And all that work shows how important human milk is. So the power of human milk is really something I try to hit home here, the first 2-3 slides. And I know you know that much better than anyone else, but I think it's important to start with that. So taking this sentence from the Lancet Breastfeeding series that the death of 823,000 children and 20,000 mothers each year could be averted through universal breastfeeding, along with the economic savings of $300 billion, is something that we are trying to understand how this works on a molecular level. And just to give you a few examples, still every single day we lose about 2,200 infants and children under the age of 5 to diarrheal disease. And we know, on the other hand, that children who are breastfed have substantially lower risk of suffering and dying from infectious diseases, including respiratory infections. And I highlight this here on purpose. And the question for us is, how does this really work? What is in human milk that makes it so powerful to save lives? At the same time, it's not just the immediate benefits, it's long-term benefits as well. We know that infants that are breastfed have lower overweight and obesity risk, lower diabetes risk, higher IQs, earning potential, and many, many other benefits for life. But we don't know exactly is how this actually works. And last but not least, there's benefits for moms as well. We know that moms that breastfeed, and the longer they breastfeed, they are at lower risk for diabetes, lower risk for cardiovascular disease, and a lower risk for breast and ovarian cancer. And once again, we don't exactly know how this works. And shortly before the pandemic hit, literally a week before the pandemic hit, we asked, this is an article that we wrote in the magazine Science here, we asked for a timely investment in better understanding the mother breast milk infant triad. So we literally called for these timely investments in this paper a week before the pandemic was called. And so this is an intro that, of course, you all know, but I think this is very important to keep in mind, that we know about the power of human milk. At the same time, when a pandemic strikes like this, what we don't know is breast milk safe? I think that's the very first question that we need to answer. And we need to answer that immediately. And you see all the consequences that come out when we don't answer this question immediately, because the uncertainty certainly led to many, many people, and Donna is going to talk about that later, to stop breastfeeding. And so why do we talk about human milk in times of a pandemic? First is the safety issue. Is the virus present in human milk and can potentially be handed over to the infant and contaminate the infant? And can the baby get infected through breastfeeding? And that is very important because we need to guide and support parents and healthcare providers about the safety of human milk. So background to when we came into this field, there were several published studies that reported the occasional presence of SARS-CoV-2 viral RNA in breast milk of infected women. And we published this here in a review article that came out in Maternal and Child Nutrition recently, where we listed all the data that was available until then for the different coronaviruses and for SARS-CoV-2 in particular. Here's one of those studies that came out. This is a paper that was published in The Lancet from a German group in Ulm. very well done. They report on 2 different moms and 2 different infants, moms co-housed together at delivery or shortly after delivery. And you see, they investigated moms and infants together, but also looked specifically when milk samples, represented here by the little bottles, tested positive for SARS-CoV-2 mRNA. And indeed, mother 2 here had several samples, sequential samples, that tested positive for the virus. And of course, that raises the big alarm bells. Is that possible that this infant actually gets infected now through breast milk? And you see that the infant indeed got infected down here, but that doesn't mean that the infection took place through breast milk. Okay, so at this point, we had several knowledge gaps. How frequent is this actually? This was 1 in 2 moms in this one study that I just highlighted. How frequent does viral RNA actually occur in breast milk? And does the presence of viral RNA represent active virus that can replicate and cause disease? So we got together with our team here in San Diego and our collaborators up in Los Angeles at UCLA. And we're in a very fortunate position that we have a human milk biorepository that had been collecting milk samples throughout North America for about 3 years already. So when the pandemic hit, we had samples from before the pandemic, but also throughout the pandemic where moms would send in samples, and it turned out that they later tested positive. So that the biorepository is led by Dr. Christina Chambers at UC San Diego down here, and together with Carrie Bertrand. And we then teamed up with UCLA Health, where Grace Aldrovandi is working, and we had already worked with her on HIV and human milk oligosaccharides in the past and published multiple papers on that. And they happen to have assays ready to go and be validated for human milk. And I want to make this very clear that you can't just take an individual assay that's used for blood or for saliva or whatever it is. You need to validate these assays for human milk. So it's very important that we do this right. Okay, then we immediately got emergency funding from the Family Larsson-Rosenquist Foundation to do some of this work, because you can imagine this work is not cheap. We also got a personal donation out of our community here from someone who had already invested money in our milk bank and immediately came to our help and supported us. We also received supplies from Medilla and guidance from the Mother's Milk Bank Association, especially the ones in Austin, Texas. Okay, so to the first knowledge gap, how frequent does viral RNA occur in breast milk? In this slide, and this is published now in JAMA as a research letter, we had milk samples from 18 different moms, and you see those different participants over in the y-axis where it says participant number, and we had multiple sample per participant, so you see that on the x-axis, and every block that we have here is a milk sample that we had available. This minus 140 means that it was 140 days before the time that the woman tested positive. Whenever you see a white dot, that is when the samples were collected during a time when mom was asymptomatic. And the black dots is when the person was actually symptomatic. Right, so we have a whole variety here of samples that were collected before the positive test. There were samples after the positive test. There were samples that were collected when mom was asymptomatic, and there were samples that were collected when mom was symptomatic. Then we tested all of these samples, 64 in total for this publication. We now have about 200 samples collected, and tested that by RT-PCR, so that is testing for mRNA fragments of that virus. And we found one sample that tested positive in this participant number 8, and it was the first sample that was collected when this person was actually symptomatic and before a positive test. So that happens quite often. A person is symptomatic, develops and doesn't get tested right away, right? So this person was probably already positive and developed symptoms, and then went eventually to the doctor and got a test and tested positive. So that's 1 out of 64, or 1 mom out of 18 participants. Now, this is all RNA. If we take a closer look at how this actually looks like on a molecular level, what are we testing there actually? And I think that is very important to keep in mind. So if we take a closer look at how these viruses look like, if you cut this open, you see outside there's a lot of these spike proteins, and then there's this envelope, and in the middle here, there is RNA. And what we test specifically with the RT-PCR is fragments of these RNAs. Right? So RT-qPCR measures RNA fragments, but that does not mean we're testing for active virus. So that is the next question that we needed to address. Does the presence of viral RNA actually represent active virus that can replicate and cause disease? And to show you this in another animation, what does it actually mean? So if you have a virus that goes in, and this is an example from another virus, but it works just as well. The virus goes into the epithelial cell and eventually multiplies in the cell and comes back out. And then it really infects the person, and it's like this multiplying event of viruses that now are in the airway and go through the entire body. So we wanted to mimic this somehow in the lab, and this is not trivial. We're working with a highly contagious virus, so we need to do this under BSL-3 conditions. And the way we do that is we take epithelial cells to mimic the epithelial cells in the airway, then take the cultured virus, and this is SARS-CoV-2 virus, add that to the epithelial cells, wait 4 days, and then measure whether that virus that we just added is actually replicating, so multiplying in tissue culture. And we can then measure that afterwards by seeing if it actually destroys the cells, which it does if it replicates, and measure the amount of viral RNA that now pops out. So you add a certain amount of viral RNA, and if it is replication competent, then your RNA will actually multiply several times. So that means that the virus was able to replicate. Okay, now this is an assay that was established before, but we needed to establish this for and validate this for human milk as well. So what we did is we took human milk that we had already collected and was in the freezer from before the pandemic. So this was from 2017. We then take the virus, add that to the milk, add the milk to the cells, wait 4 days, and see if there is replication. So this is to validate an assay if a virus was in human milk. If there is indeed replication competent virus, can we measure that in tissue culture? And the answer is yes. So we see that the cells get destroyed, and we see that the virus replicates. So the assay works. Now what we do is we test this in our actual samples that we collected during the pandemic, including the sample that tested positive for viral RNA. So we take that milk, these milk samples, add that to the tissue culture, wait 4 days, and see what happens. Evaluate the cells and measure the RNA. And the result here is that none of these milk samples, none of the 64 milk samples that we had collected at this point, including the one that had tested positive for RNA, showed any sign of replication competent virus. So there was no virus measured afterwards. So the presence of viral RNA alone is not the same as the presence of replication competent virus. Just because we measure a fragment of the virus doesn't mean that there's an intact virus in milk that can cause disease. Okay, in addition to that, we also tested, is it possible that if the virus ever gets into milk, for example, through human donor milk processing, would Holder pasteurization inactivate it? So here what we did, again, we took SARS-CoV-2, added it to human milk from 2017, then used Holder pasteurization to treat the milk, added the milk to the cells, and once again, after 4 days, we did not find any replication competent virus in the samples. So in other words, Holder pasteurization inactivates SARS-CoV-2 in breast milk should it ever get in there. So even if breast milk was contaminated with SARS-CoV-2 during pumping and handling, Holder pasteurization, which is commonly used by human milk banks, inactivates the virus in contaminated breast milk. So summary of this short study is that the presence of SARS-CoV-2 RNA in breast milk of infected women is extremely rare. We found it in 1 out of 18 women published in this paper, and 1 out of 64 samples altogether from these 18 women. We've now recruited and analyzed about 50 women and 200 different milk samples, or a little over 200 different milk samples, and the stats look about the same. And very important, the presence of viral RNA is not the same as presence of active virus. And last but not least, Holter pasteurization inactivates the virus should breast milk ever get contaminated. So transmission of SARS-CoV-2 from mother to infant through breast milk is highly unlikely based on the data that we collected, and others have confirmed this in the meantime. Now, on the other hand, now that we established safety, which is absolutely key, now, is there anything in human milk that is actually having antiviral activities? So are there components in human milk that can stop the virus? For example, antibodies. So mom gets exposed, has the virus, makes antibodies, they show up in the blood. Are they transferred now into the milk and potentially provide protection to the infant as well? We study a lot oligosaccharides in my lab, and we know that some of them have antiviral activity for other viruses. Is it possible that oligosaccharides also have antiviral activity against SARS-CoV-2? And then there's many other components in human milk, including antimicrobial peptides and many other things that could potentially have antiviral activity. And again, that is highly important if we want to give actual data to guide and support parents and healthcare providers to show that human milk is not only safe, but it actually provides a huge benefit that we already know for many other things, but also specifically in the context of COVID-19. And last but not least, is it possible to then take some of those components that we find in human milk to be efficient, synthesize them outside of human milk? I'm not saying that we want to now take human milk and give it to everyone that has COVID-19, but can we synthesize some of those components based on the knowledge that we just generated and use that as novel antiviral components against COVID-19 for people of all ages to really stop this pandemic and highlight really the power of human milk and human milk components as we use them in a different context in adults, for example. Okay, what does the success look like? What does success look like? We want parents to be confident to breastfeed. And at the very beginning of the pandemic, and again, Donna Geddes will talk about this, and still there is a lot of uncertainty about the safety of human milk. And we need actual data to show and guide the health organizations to say, look, we have the data, human milk is safe, people need to continue breastfeeding. We want healthcare providers to make recommendations based on evidence, based on science, not based on fear, not based on dogma, but on actual data. And ideally, we would like to get novel and safe antiviral components to prevent and treat COVID-19 in all age groups. So really learn from the power of human milk and develop new therapeutics that we can use in the fight against this virus. And last, but certainly not least, I think what this pandemic really has shown us is that we are not ready when a pandemic like this strikes. That is true for so many different things in our society, but certainly also for the breastfeeding context. We can always say, breast is best and let's continue breastfeeding, but we need to evaluate the safety first. Otherwise, if there is ever a virus or ever a pathogen that gets into human milk and does indeed transmit to infants, we need to know that, and we need to know that based on data. Okay, so I'm coming back to this statement, and I add to that we have to evaluate the risk of SARS-CoV-2 transmission through breast milk, but we already know the risk associated with not breastfeeding. So please always keep that in mind, that yes, we can push breastfeeding because we know the risks that are associated with not breastfeeding, but at the same time, we need to make sure that breast milk is safe, and we need to make sure that that's actually based on data. Like I mentioned, I think there's great opportunities for people of all ages, and there is an opportunity to harness the power of human milk to discover new antiviral components that potentially help us in this pandemic and maybe in other pandemics as well. And I'll leave you with that and take questions if there are any.